Today, we are going to look at something that is hiding in plain sight. I mean, it is literally inside every single one of your cells. It is on the surface of your cells. It is floating around in your blood right this second.
Right now.
But if I said the word sugar to you, just reflexively, what is the very first thing that pops into your head?
Oh, it has to be energy, right?
Yeah.
We think of glucose. We think of, fueling the machine.
Yeah.
Or if you've been looking at nutrition labels lately, you probably think of calories, insulin spikes, all the things you're sort of supposed to avoid.
Exactly. That is the cultural baggage of the word sugar. We think of it as fuel, or we think of it as a treat, maybe something bad.
Yeah.
But the stack of research papers and textbook chapters we are unpacking today, specifically focusing on Chapter 46 about glycoproteins, it flips that idea completely on its head.
It really does.
It turns out that in the context of cellular biology, sugar isn't just fuel.
Oh.
It's information. It's a language.
That is the perfect way to frame it. I mean, if you look at the biology of the cell, sugars, and we're talking about these complex chains called oligosaccharides, they are acting like a sophisticated barcode system.
A barcode system.
Yeah, or a high-density coding system that cells use to talk to each other, to organize themselves into tissues, and crucially, to defend themselves against invaders.
And we aren't just talking about, some biological trivia here. This sugar code is absolutely central to everything. We're going to talk about how it dictates your blood type, how viruses like the flu and even HIV actually break into your body, and even how a sperm cell manages to find and fertilize an egg.
It's all sugar.
It's all sugar.
It really is. And the technical term for this, the star of our show today, is the glycoprotein.
Glycoproteins. Okay, so let's start with the basics, but I want to go deep immediately. Definition time.
Let's do it.
What exactly are we looking at when we say glycoprotein?
Simply put, a glycoprotein is a protein that has carbohydrate chains attached to it. These chains are called glycans. And the key here, this is really important, is that they are covalently bound to the amino acids that make up the protein.
Covalently bound. So this isn't just like a sticky situation where a sugar molecule bumps into a protein.
Not at all. They are chemically welded together. It's a permanent modification.
So it's a hybrid molecule. It's part protein, part sugar.
Correct. And this isn't some rare occurrence or an exception to the rule. This is actually the most frequent post-translational modification of proteins in the human body.
Okay, post-translational modification. Let's unpack that jargon for a second.
Yeah, good idea.
We know translation is the step where the instructions from your DNA are turned into a protein chain.
Right, in the ribosome.
So this is something that happens after the protein is already born.
Exactly. The DNA gets read, the protein is built, that's the translation, but the protein isn't finished yet. It's like an undecorated Christmas tree.
I like that.
The cell then modifies it, it decorates it, and in this case, it decorates it with sugar.
And you're saying this is the most common decoration?
By far. To give you a sense of the scale, almost all the proteins circulating in your blood plasma right now are glycoproteins.
Wow.
A huge number of the proteins embedded in your cell membranes are glycoproteins. Many of your hormones, your antibodies, they're all glycoproteins.
So if I could shrink myself down, magic school bus style, and stand on the surface of a human cell, I wouldn't be standing on a smooth, polished balloon like in the textbooks.
Not at all. That classic diagram from high school biology is it's wrong. It's a massive oversimplification.
Okay, so what would I see?
If you were standing on a cell, you'd be standing in a dense forest. Or maybe a coral reef is an even better analogy.
Like a coral reef.
Yeah, the surface of a cell is covered in these waving, branching chains of sugars sticking out into the extracellular space. Scientists have a name for it. The glycocalyx.
The glycocalyx.
It literally means sugar coat.
A sugar coat. I love that image.
Yeah.
But unlike the sugar coating on a donut, which is, there to make it taste good,
right,
this coat is doing some serious heavy lifting.
Tremendous lifting. I mean, it's checking IDs, it's signaling to neighbors, it's providing structural support, it's protecting the cell membrane.
And if you mess up the code.
If you put the wrong sugar in the wrong place or miss a sugar completely, the consequences are catastrophic.
We're not talking about a minor glitch.
Not at all. We're talking about the complications from diabetes, how cancer metastasizes, things like cystic fibrosis. It's very often the difference between health and severe disease.
Okay, let's get into this code. If this is a language, we need to know the alphabet.
Right.
When we talk about these sugars being attached to proteins, are we talking about the sucrose in my coffee cup?
No, and this is where it gets fascinatingly specific. Nature is very, very picky.
Okay.
There are tons of different monosaccharides in the world. Plants make all kinds of weird and wonderful ones, but in human glycoproteins, only 8 predominate.
Only 8. That seems, I don't know, kind of insufficient. How do you build an immune system and a brain and everything else out of just 8 Lego bricks?
That's the beauty of combinatorics. I mean, think about it. The English language has 26 letters.
Right.
DNA has only 4 bases, A, C, T, and G. And with just those 4, you get all of life.
That's a good point.
The sugar alphabet has 8 letters. But here's the kicker. Unlike DNA, which is just a linear string, beads on a necklace.
A to T, C to G.
Exactly. Sugars can branch. They can link at different carbon atoms. They can form these incredibly complex 3-dimensional structures.
Ah, so it's not just the sequence, it's the shape.
It's the shape. With just those 8 sugars, the number of possible words or instructions you can write is exponentially higher than with amino acids or nucleic acids. It's a massive information-dense system.
Okay, I'm sold on the potential. So who are the 8? I'm assuming our old friend glucose is in there somewhere?
Yes, of course. You have the familiar ones, glucose and galactose. They're isomers of each other, very common.
Okay.
Then you have mannose, and we will see that mannose is absolutely crucial for the structural core of a lot of these things.
Mannose. Got it.
And then it gets a bit more chemically complex. You have fucose.
Fucose with an F.
With an F, yes. Fucose.
Yep.
And you have xylose.
Okay.
And then there are 3 amino sugars. Now, these are sugars that have an amino group attached, which changes their chemistry pretty significantly.
Right.
You have N-acylglucosamine.
That is a mouthful.
It is, which is why we all just shorten it to GLC-NAC, GLC-NC.
GLCN. Okay.
Then you have its twin, N-acylgalactosamine, or GalNAc.
GLCNAC and GalNAc. They felt like a pair of alien twins in a sci-fi movie.
They kind of act like it, too. They are structurally very, very similar, but they play completely different roles, which we'll get into.
And the 8th one.
The 8th one is the most distinct, the VIP of the group, N-acetylneuramic acid.
Which we know by a much cooler name.
Yes, sialic acid, or NUAC for short.
I want to pause on sialic acid because the source material really seemed to suggest this one is special.
Oh, it is.
It's not just another brick in the wall.
It is very special. For one, it's the only one of the 8 that carries a net negative charge at physiological pH.
And that's a big deal in chemistry.
A huge deal. It changes how the entire molecule interacts with water, with ions, with other molecules. It's like putting a static charge on the end of a wand.
And its position is also important.
Critically important. Sialic acid almost always sits at the very end of the sugar chain. It's the terminus.
So it's like the period at the end of the sentence.
Or maybe a better analogy is it's the flag at the top of the mast.
Okay, I like that.
Because it's at the end of the chain sticking out the furthest. It is the first thing that other cells or molecules encounter when they bump into that glycoprotein.
So it's the handshake.
It's the handshake. It can repel things because of that negative charge, which is why your red blood cells don't all stick together in a big clump.
Oh, okay.
But it also acts as a very specific docking site for receptors, both our own and, as we'll see, for viruses.
Okay, so we have these 8 sugars, and the cell springs them together in these branching patterns to create specific shapes or signals.
Right.
But here is something that really confused me in the reading. We usually think of biology as being super precise. DNA is perfect. A pairs with T, C pairs with G. It's digital.
It's a template.
Right. But the text mentions something called microheterogeneity.
Well,
and it sounds like the cell's being a little bit sloppy.
sloppy is one way to look at it, but a biologist would call it versatile or dynamic.
Okay.
You see, DNA as a template, it's a hard master. Every single time you read a gene, you get the exact same amino acid sequence for a protein. It's a perfect photocopy.
1-to-1.
But glycosylation, the adding of the sugars, is a secondary process. It is
Right.
not template-driven in the same way.
So how does it work then? If there's no template, how does the cell know what to build?
It depends on the environment in the factory floor of the cell, in the ER and the Golgi. It depends on which enzymes, specifically these enzymes called glycosyltransferases, are available at that moment to attach the sugars.
So it's about what tools are on the workbench at that second.
Exactly, and it depends on the availability of the sugars themselves. So it's a much more probabilistic or stochastic process.
So you could have the exact same protein molecule, let's say a hormone like EPO,
Classic example.
but one version of it that was made at 10:00 a.m. might have a slightly different sugar chain than the version that was made at 10:05 a.m.
Precisely. That is microheterogeneity. You have the same polypeptide backbone, but a whole population of variable glycoforms.
Why would nature do that? I mean, isn't variability bad when you're trying to run a tight ship?
Not necessarily. In fact, this variability allows for massive complexity and fine-tuning.
How so?
It allows the cell to modulate how that protein interacts with the world without having to change the underlying genetic code. It's a much faster way to adapt.
So it's like having a standard uniform for an army,
Yeah.
but being able to swap out the badges and the patches depending on the specific mission, or even depending on how many badges you had in stock that morning.
That is a fantastic analogy. It gives the system incredible flexibility.
So the protein is the soldier, and the glycans are the rank and the insignia.
Exactly. And sometimes changing that insignia changes the soldier's orders completely.
Okay, so we've got the ingredients, we know about the variability. Now let's look at the actual structures. The source material breaks glycoproteins down into 3 main classes based on how these sugars are actually glued onto the protein.
Right.
And I think it's important we visualize these because they look and act really differently.
Absolutely. The classification is all determined by which amino acid in the protein the sugar chain grabs onto. The first major class is O-linked glycoproteins.
O-linked, and the O stands for oxygen.
Yes, exactly. The sugar chain links to the hydroxyl group, the oxygen atom of the amino acids serine or threonine.
Serine and threonine. Got it. And what do these typically look like? Are they long, spindly things?
Think hairy, or even better, think of a bottle brush.
A bottle brush, like the kind you use for cleaning baby bottles.
Exactly that. A classic, and I mean the textbook example, of O-linked glycoproteins are the mucins.
Mucins, as in mucus.
Yes, the very proteins that make up the mucus in your respiratory tract, your digestive tract, your reproductive tract.
Okay.
These mucin proteins have regions in their sequence called tandem repeats that are just absolutely densely packed with these O-linked sugars.
So the protein is the wire in the center of the brush.
The central core.
And the sugar chains are the bristles sticking out in every single direction.
Precisely. And now imagine millions and millions of these bottle brushes packed together. They trap a huge amount of water. They become slimy, viscous. They take up a huge amount of space.
And what is the functional point of being a bottle brush? I mean, why would the cell spend all that energy making sugars just to look like a cleaning tool?
Protection and lubrication. It's a physical barrier. Because there are so many sugars packed so tightly together, the molecule becomes very rigid and extended. It can't fold up. It holds water, creating that gel-like mucus layer, but importantly, it is incredibly resistant to digestion.
Which makes sense. If you're lining the stomach, you don't want to get digested by your own stomach acid and proteases.
Exactly. The density of those O-linked sugars physically protects the protein backbone from proteases, the enzymes that cut up proteins. It's a shield. It keeps bacteria off your actual cells. It keeps acid away from the lining. It's a slimy, sugary shield.
And if you strip those sugars away,
the protein backbone would be eaten up in seconds.
Okay, so O-linked is the bottle brush shield. What's the second class?
The second class is N-linked glycoproteins. And here, as the name suggests, the sugar attaches to the nitrogen of the amino acid asparagine.
N for nitrogen. That's easy enough to remember.
It is. But it's not just any asparagine. This is a key detail.
Okay.
It has to be part of a specific code sequence in the protein. Asparagine, then X, where X can be almost any amino acid except proline, and then serine or threonine.
As an Xer or as an XDHR. It's like a secret password.
It is. It's a consensus sequence. The enzyme that does the transfer, it scans the protein looking for that password. If it sees it, it knows, okay, stick a sugar here.
And do these look like bottle brushes too?
No, they're quite different. N-linked chains are generally more complex, more branched. They act less like a physical shield and more like antenna or sensor arrays.
For communication.
For communication, for recognition. And interestingly, all N-linked glycoproteins share a common ancestor, structurally speaking. They all start with the same pentasaccharide core.
Pentasaccharide, meaning 5 sugars.
Yes, that's right. 3 mannoses and 2 GLCNX deep down near the protein surface. No matter how weird and complex the branches get later on, whether it becomes what we call a complex type or a high mannose type, they all have that same root structure.
That implies a very, very conserved evolutionary history.
It does.
It's like nature found this one way to attach sugar to nitrogen 1 billion years ago and said, okay, if it ain't broke, don't fix it.
It absolutely suggests this system was developed very early on and is essential for most eukaryotic life. From that core, the cell can then build this massive tree of possibilities for cellular recognition.
Okay, so we have O-linked bottle brushes and N-linked antenna was the third one. The source mentioned it was a bit of an outlier.
The third one is a bit of a rebel, yeah. It's the GPI-anchored protein.
GPI stands for, let me check my notes here, glycosylphosphatidylinositol.
You got it.
That is a word score in Scrabble right there.
It is. But the concept is actually cooler than the name. Normally, proteins that sit in the cell membrane are transmembrane.
Right, they go all the way through the membrane like a stud in a wall. They're stuck there.
They're pretty fixed in place. But GPI-anchored proteins are different. They aren't drilled through the membrane. They are tethered to a lipid tail, the GPI that just sits in the outer leaflet of the membrane.
So they aren't drilled in. They're just floating on top, attached by a leash.
That's a great way to think of it. They are anchored, but the anchor is in a fluid lipase layer, and this gives them very high lateral mobility.
They can slide around.
They can slide around the surface of the cell much, much faster than a transmembrane protein can.
It's the difference between a raft floating on a lake and a pier that's been driven into the mud at the bottom.
Perfect analogy. And this mobility is crucial. It allows these proteins to quickly cluster together into these lipid rafts for signal transduction, for moving messages across the cell surface very quickly. Or it allows them to be rapidly cleaved off if the cell needs to release them into the environment.
Okay, so we have our structures. We know what they look like. But now I want to get into the weeds of the mechanics of construction.
The factory floor.
The factory floor. How on earth does a cell build these things? I mean, building a protein is already hard enough. Transcribing DNA, translating RNA, but now you have to decorate it with these complex sugar trees in very specific patterns.
We have to go to the endoplasmic reticulum, or ER, and then the Golgi apparatus.
The classic organelles from biology class.
The protein synthesis and processing pathway. But the process is fundamentally different for the two main types we just discussed.
O-linked versus N-linked.
Right. O-linked sugars are usually built step by step, one sugar at a time, directly onto the protein, usually in the Golgi. It's like stringing beads on a necklace.
Simple enough.
But N-linked sugars, they do something truly wild. They are prefabricated.
Prefabricated, you mean like a modular home?
Exactly like a modular home. The cell doesn't build the tree on the protein. It builds the entire core sugar tree, all 14 sugars of it separately, on a special carrier molecule.
A carrier?
Yes, a massive lipid called dolichol.
Dolichol. The source material describes this as being just huge.
It is enormous. It's a polyisoprenoid made of 17 to 20 isoprene units. It's this long hydrophobic molecule that's buried in the membrane of the ER.
And its job is?
Think of dolichol as a forklift or a workbench. The cell builds the entire 14 sugar chain on the dolichol, one sugar at a time. And then, in a feat of molecular gymnastics, it flips the dolichol with the entire sugar chain attached from the outside of the ER membrane to the inside.
Hang on. You just breeze right past, flips it.
Laughs. It's a big deal.
We're talking about a massive lipid carrier holding a big polar tree of sugars. And it just, it somersaults inside the membrane.
I'm glad you caught that because it is a thermodynamic nightmare. Membranes are stable. They do not like things flipping back and forth. This move translocating the dolichol sugar complex from the cytoplasmic side to the internal lumen side is a major, major feat of cellular engineering.
It sounds like trying to flip a pancake while the pan is inside a zipped-up backpack.
That is a surprisingly accurate image. The cell has to use dedicated proteins, colloquially called flippases, just to manage this incredible gymnastic move.
But why? Why go through all that trouble?
Because here's why it matters. The raw materials for building the sugar tree, the sugar nucleotides, are in the cytoplasm on the outside of the ER.
Okay.
But the protein itself is being born and threaded into the inside of the ER, into the lumen.
So the flip is the bridge. It connects the supply depot on the outside with the assembly line on the inside.
Exactly. And once it flips, that sugar tree is committed. An enzyme called an oligosaccharyl transferase transfers that entire pre-made sugar block, all 14 sugars at once, from the dolichol onto the waiting asparagine of the protein.
Wham! Stamped on.
Stamped on. And block transfer. It's incredibly efficient, but it's also very high stakes. If that tree is built wrong on the dolichol, every protein getting stamped that day is going to be defective from the start.
That is just incredible engineering. But hearing about all this complex assembly, it makes me worry. What happens if something goes wrong?
It does go wrong.
What if the sugar is put on crooked? Or the protein itself folds weirdly after it gets the sugar? Is there any quality control?
This brings us to one of the most elegant quality control systems in all of biology, the Kalnexin cycle. I like to call this the school of folding.
I love this part of the reading. This is where the cell essentially inspects its own work. Walk us through it.
So you've just stamped this 14 sugar chain onto your new protein. Now the protein has to fold into its correct 3D shape, but folding is hard. It needs help.
Right.
In the ER, there are chaperone's helper proteins called calnexin and calreticulin.
Calnexin and calreticulin, they sound like Roman sentinels.
They absolutely act like it, and importantly, they are lectins.
They bind sugar.
They bind sugar. Specifically, they recognize a very specific stage of that sugar chain we just added. They look for a chain that has exactly one glucose molecule left on it.
Just one.
Just one. They grab the protein by that single glucose handle and they hold on to it.
And why hold it? What's the point?
To keep it from clumping together with other unfolded proteins and to give it a safe, protected space to try and fold correctly. They're like spotters in a gym. They hold the weight while the protein gets its posture right.
And then what happens?
Once the protein has had some time to try and fold, another enzyme comes along and snips off that last glucose. The handle is gone. Calnexin lets go. The protein is now free.
And if it's folded correctly at this point?
If it's folded correctly, it's allowed to leave the ER and head on to the Golgi for finishing touches. It's graduation day.
But, and this is the crucial part, what if it's still folded wrong? What if it's just a tangled mess?
This is the genius part of the system. There is a sensor enzyme, a glucosyltransferase, that floats around the ER. Its only job is to bind to misfolded proteins.
It's an inspector.
It's an inspector. And if it sees a naked misfolded protein, it reattaches the glucose.
It puts the tag back on.
It puts the tag back on, which means calnexin grabs it again.
So it sends it back to school.
It sends it back to school. Try again, fold again. It enters this cycle of binding and release.
That is amazing. It's a loop. It just keeps recycling the protein until it gets it right.
Well, to a point. If a protein fails repeatedly, eventually the cell loses patience. A different enzyme slowly trims off some of the mannosugars in a specific way that marks the protein for destruction.
It's a timer.
It's a timer. Once that mannose signal is there, the protein is exported out of the ER and fed to the proteasome, which is essentially the wood chipper of the cell.
That is ruthless but necessary efficiency.
It has to be. Misfolded proteins are incredibly dangerous. They can aggregate and cause diseases like Alzheimer's or Parkinson's. This sugar-based quality control system ensures only quality products leave the factory.
We mentioned the word lectins just now.
Yeah.
Calnexin is a lectin. I feel like lectins are a buzzword in diet culture these days, usually in a negative way.
Oh, yeah. Don't eat beans, they have lectins.
Right. But in biochemistry, what exactly is a lectin?
We need to reclaim the word. In biochemistry, a lectin is simply any protein that binds to a carbohydrate. That's it. It's a decoder. It reads the sugar code.
So antibodies bind to antigens?
And lectins bind to glycans.
Okay. And they are everywhere.
Everywhere. They were first discovered in plants. That's the bean connection, phytohemagglutinin. But animals, including us, rely on them for everything. We just saw calnexin, which is an intracellular lectin. But there are also lectins on the surface of your cells and floating in your blood.
The source material mentions the liver has a really specific and clever way of using lectins to check the expiration date of proteins in the blood. Can you explain that? It sounded almost like a recycling center.
This is the Asia-Glycoprotein receptor. It's a mouthful, but the concept is just beautiful.
Okay.
Many of the proteins in your blood plasma have these complex sugar chains ending in sialic acid. Remember, that's the flag at the end of the chain.
Right, the terminal cap with the negative charge.
Exactly. Now imagine a protein that's been floating around in the turbulent bloodstream for days or weeks. It gets banged up.
Wear and tear.
Right. And over time, those sialic acid caps can get knocked off or degraded by enzymes in the blood. When the sialic acid is gone, it reveals the sugar that was sitting just underneath it, which is usually a galactose residue.
So the sialic acid is like a protective seal covering the galactose.
It is. Now, the cells in your liver, hepatocytes, have these receptors on their surface that are specifically shaped to look for exposed galactose. They're galactose-binding lectins.
So they're just scanning the blood for this signal.
Constantly. If they see a protein with an exposed galactose, they bind to it, pull it out of circulation through endocytosis, and send it to their own lysosomes to be destroyed and recycled.
Wow. So the sialic acid is essentially the freshness seal. And if that seal is broken, the liver assumes the protein is old and damaged, and it just throws it in the trash.
That's exactly what it is. It's a biological timer mechanism that's built right into the chemistry of the molecule itself. It ensures your blood is constantly being refreshed with new functional proteins.
That is brilliant. It's planned obsolescence, but for a good cause.
Exactly.
But Looking at our outline here, we're moving into the when it goes wrong segment. And it seems like when this elegant sugar machinery breaks, the diseases that result are pretty terrifying.
They are, because these processes are so fundamental, happening in every single one of your cells. The defects are often systemic and absolutely devastating. A classic example is eye cell disease.
Eye cell, which stands for inclusion cell disease.
Right. This is a perfect example of a failure in address labeling inside the cell.
Address labeling.
So inside your cells, you have organelles called lysosomes. Think of the lysosome as the stomach of the cell. It's full of acid and dozens of powerful enzymes designed to digest waste and recycle old parts.
Garbage disposal unit.
The garbage disposal. Now, the enzymes that work inside the lysosome are proteins. They're made in the ER and processed in the Golgi, just like all the other proteins we've been talking about.
Okay.
But how does the cell know to send this specific protein to the lysosome and not, say, secrete it out into the blood?
It needs a shipping label, a tag that says, to lysosome 123 cell street.
And that label is a sugar. Specifically, it's a modified sugar called mannose-6-phosphate, M6P. A specific enzyme in the Golgi recognizes these lysosomal enzymes and adds a phosphate group to one of their mannose sugars. Then, a receptor in the Golgi sees that M6P tag and directs the cargo into a vesicle bound for the lysosome. A very precise sorting system. So what happens in eye cell disease? The patient has a genetic mutation in the enzyme that adds the phosphate. They can't make the label.
So the garbage men, they don't know where to go.
The enzymes themselves get made perfectly. They're functional. But because they lack the M6P tag, the cell's sorting system treats them as default cargo and secretes them outside the cell into the blood.
They're lost in the mail.
They're lost in the mail. But the real tragedy is what's happening back inside the cell. The lysosome, the garbage disposal is now empty of enzymes. It can't digest anything.
So the trash just piles up.
It piles up rapidly. The cell fills up with undigested glycosaminoglycans and lipids. These huge bloated vesicles full of junk are called inclusions.
And inclusion cell disease.
Exactly. And the clinical result is devastating. Severe psychomotor retardation, skeletal deformities, coarse facial features. Patients usually die in the first decade of life.
All because of a single missing phosphate on a single sugar chain.
It's a tragic illustration of how critical the sugar code is. Just one tiny chemical error in the code and the entire system backs up.
Let's talk about another one from the list. PNH, paroxysmal nocturnal hemoglobinuria.
Another fascinating one.
I want to break that name down because it sounds like something out of a 19th century ghost story.
It does.
Proximal means sudden attacks. Nocturnal means at night. Hemoglobinuria means hemoglobin in the urine.
Which means your red blood cells are bursting.
Right. But the nocturnal part is what gets me. Why would your red blood cells decide to burst, specifically at night?
It really highlights how incredibly fragile our biochemistry can be. Think about what happens when you sleep. Your breathing pattern shifts.
It gets shallower.
It gets shallower. You aren't exhaling CO2 quite as aggressively as when you're awake and moving around.
So you retain a little more CO2.
A tiny bit more. And CO2 in the blood turns into carbonic acid. So when you sleep, your blood becomes marginally more acidic. We're talking a tiny pH shift, totally normal for everyone, but
There's a but.
there's a but. Your immune system, specifically this ancient part of it called the complement system, is extremely sensitive to pH. It gets just a little bit more trigger happy in slightly acidic environments.
Okay, so the immune system gets a little more aggressive while you sleep, but why does it attack your own red blood cells?
Normally it doesn't, because your red blood cells have shields. They have proteins on their surface called DAF, which is decay accelerating factor, and CD59. Their job is to tell the complement system, hey, don't shoot, I'm one of the good guys.
Friendly fire protection.
Exactly. But here's the catch. DF and CD59 are both GPI-anchored proteins. Remember the floating anchor.
The leash and the lipid layer.
Right. In patients with PNH, they have a genetic mutation that prevents them from building the GPI anchor itself. So they make the DIF protein just fine, but it has no way to attach to the cell surface. It just floats away.
So the red blood cell is naked. It has no ID badge to show the immune system.
It has no shield. And so your own immune system, specifically that complement system that's a little more active at night due to the pH shift, attacks it. It punches holes in the red blood cell membrane, causing it to lyse or burst.
And the patient wakes up?
And their urine is dark because it's full of hemoglobin from all the destroyed blood cells.
That is just wild. The chemistry of sleep triggers the immune system to attack your own anchorless cells. It's a perfect storm of defects.
It's a fascinating, albeit terrible, intersection of physiology and biochemistry.
Let's switch gears to a disease that affects millions of people, not just a rare few. Diabetes.
Mm-hmm.
We all know diabetes is about having high blood sugar. But the source material makes a really, really critical distinction between glycosylation and glycation.
This is probably one of the most important practical takeaways from this entire deep dive.
Yeah.
Glycosylation is what we've been talking about this whole time. Enzymes carefully and precisely building specific sugar structures for a biological purpose. It is controlled. It is good.
It's the intended process.
And glycation. Glycation is chaos. It is non-enzymatic. It is what happens when you have way too much sugar floating around in your blood and it just randomly, spontaneously smacks into proteins and sticks to them.
So it's like if you spilled syrup on your laptop keyboard.
Yeah.
It's not supposed to be there, and it just gums up the keys.
That is the perfect analogy. And in diabetes, when blood glucose is chronically high, this is happening everywhere in your body. A prime example is hemoglobin A1C.
HbA1c. I think everyone who has had a blood test has seen that on the chart.
Right. HbA1c is simply a hemoglobin molecule that has been glycated. A glucose molecule has gotten stuck to the N-terminal vein of the hemoglobin chain.
And because red blood cells live for about 120 days.
Exactly. Measuring the percentage of your hemoglobin that is glycated gives your doctor a record of how much sugar has been floating around for the last 3 months.
So it's not just a snapshot, like a finger prick test. It's a history book of your blood sugar control.
Yes. But the damage goes way beyond just hemoglobin. This random sugar binding leads to the formation of something called AG's advanced glycation end products.
AGs, which is aptly named because they literally age you.
Very much so. These AGs cause chemical cross-linking between proteins. They make your collagen stiff. Imagine a fishnet that gets all tangled and glued together.
So it loses its flexibility.
It loses its flexibility and function. This is why diabetics get arterial stiffening, kidney damage, and retinal damage. The tissues literally get caramelized over time.
Caramelized. That's a really visceral image. You mean like browning meat in a pan?
Chemically, it is very similar to the Maillard reaction, the browning of a steak or the crust of bread. It is, in effect, slow cooking your internal organs.
Wow.
That is why sugar toxicity is so dangerous over the long term. It's not just about calories, it's about the slow, irreversible chemical modification of your body's structural proteins.
Okay, that is sobering. Internal slow cooking is definitely going to stick with me. Let's pivot to something a bit more life-affirming.
Please.
The source material calls these biological miracles, and the first one is the ultimate miracle: fertilization.
This is one of the coolest examples of the sugar code in action. It addresses a really fundamental question. How does a sperm know it has found an egg?
Right.
And how does the egg ensure it's only fertilized by a sperm from the same species? So a human sperm and not, say, if you were swimming in the ocean, a fish sperm.
I always just assumed it was size. Or maybe just some chemical gradients.
It's glycoproteins, specifically the sugar coat on the egg. The human egg is surrounded by a thick, clear shell called the zona pellucida.
The zona pellucida. Sounds like a nebula in space.
It's a beautiful glycoprotein matrix. And one of the key proteins in this shell is called ZP3, zona pellucida protein 3. ZP3 is covered in very specific O-linked oligosaccharide chains.
The bottle brush sugars are back.
They're back, and they're playing a critical role. The sperm has a receptor protein on its head that is shaped to match that specific sugar chain on ZP3 perfectly. It's a lock and a key.
The sugar is the keyhole.
The sugar is the keyhole. When the sperm binds to the sugar on ZP3, that binding event triggers a massive reaction, the acrosomal reaction.
That's the warhead on the sperm exploding, right? It releases enzymes?
Essentially, yes. The sperm releases a payload of enzymes that allow it to bore a hole through the shell so it can fuse with the egg membrane. But here's the kicker. If you experimentally remove those specific sugars from ZP3, or if you change the sugar structure even slightly, the sperm will just bounce right off.
It won't bind.
It won't bind, it won't trigger the reaction, no fertilization.
So the very beginning of every single human life depends on a sugar handshake.
100%. And if the sugar count doesn't match, fertilization is physically impossible. This acts as the species barrier. A mouse sperm doesn't have the key for the human sugar lock.
That is just incredible. Now, the other miracle mentioned is how our immune system fights infection. Specifically, how our white blood cells get out of the bloodstream and into the tissue. the rolling mechanism.
This is one of my favorite biological mechanisms to visualize because it's so physical.
Okay.
Imagine a white blood cell, a neutrophil, rushing through one of your blood vessels. The heart is pumping, the blood flow is fast. It's like a car on a highway going 100 miles an hour.
And let's say there's an infection in your finger. So that car needs to take the exit ramp to get to the finger tissue.
Exactly. But there are no brakes on the car. The blood flow is way too fast. If it tried to just grab onto the wall of the blood vessel, the sheer force of the blood would rip it apart.
So how does it stop?
It uses Velcro.
Velcro.
The cells that line your blood vessels, the endothelial cells near the site of an infection, they get a signal to put up little flags called selectins. These selectins are lectins. They bind to sugar. The white blood cell, meanwhile, is covered in its own glycoproteins that display specific sugars, sialylated and fucosylated sugars, to be precise.
So the white blood cell is the fuzzy side of the Velcro, and the vessel wall is the hook side.
That's a great way to put it. As the white blood cell flies by, the selectins on the wall grab onto these sugars.
Does that stop it dead?
No, and that's the key. The bond isn't strong enough to stop it instantly against that rushing blood. It grabs, it pulls, the bond breaks, then another one down the line grabs, it pulls, that bond breaks.
It creates a tumbling motion.
A rolling motion. The cell slows down, it tumbles along the vessel wall, tumbling, until finally it's moving slowly enough for other, much tighter adhesion molecules called integrins to lock it in place.
And then it can squeeze through the wall.
Then it squeezes through the wall to go fight the bacteria.
That is such a cool image. It's like a plane landing on an aircraft carrier, catching the arresting wire, but doing it over and over again in slow motion until it stops.
Exactly. And without that initial weak sugar-selectin interaction, the white blood cell would just zoom right past the infection. It would never be able to get out of the highway.
And this isn't just theory. This is the cause of another one of these rare diseases.
Right.
Leukocyte adhesion deficiency.
Yes, LAD2, or leukocyte adhesion deficiency type 2. In this disease, the patient has a defect in a transporter for fucose. They can't add fucose to the surface of their white blood cells.
So the selectins on the blood vessel wall have nothing to grab onto.
Nothing. The white blood cells can't roll, they can't slow down, they can't stop.
So what happens to the patient?
The patient has incredibly high white blood cell counts in their blood. The army is mobilized and ready to fight, but they get terrible recurrent bacterial infections in their tissues because the soldiers can't get off the bus.
It's a traffic control failure at the molecular level.
Perfectly put.
All right, we've talked about how our own body uses these sugars.
Yeah.
But we aren't the only ones who know this code. The source material has a whole section on the invaders.
Hacker.
The hackers. Let's talk about the flu. Influenza. We always hear about H1N1, H5N1. I think most people just tune those letters and numbers out. What do the H and the N actually stand for?
They stand for the two major glycoproteins on the surface of the virus itself. H is for hemagglutinin, N is for neuraminidase.
I've heard those names, but I never knew what they actually did.
Think of hemagglutinin, the H protein, as the virus's grappling hook.
Okay.
It is a lectin that binds to sialic acid on the surface of your respiratory cells. That's how the virus gets in. It recognizes the specific linkage of sialic acid on your throat or lung cells and latches on.
So it hacks our sialic acid code to use as a docking station.
It exploits it, exactly. So the virus gets in, it replicates, it makes millions of new copies of itself inside your cell. Now, these new viruses need to leave to go and infect other cells, but there's a problem.
What's the problem?
As they bud out of your cell membrane, the surface of that membrane is still covered in the same sialic acid they used to get in.
So they get stuck on their way out.
They get stuck on the cell they just killed. Their own grappling hook, the hemagglutinin, grabs onto the cell surface again. They are tethered to the sinking ship.
That's a major design flaw for a virus that wants to spread.
It would be, but the virus has a solution. That's where neuraminidase, the N protein, comes in.
What does it do?
Neuraminidase is an enzyme that cuts sialic acid. It acts like a pair of molecular scissors. It snips the sialic acid off the cell surface, releasing the virus so it can float away and find a new victim.
So H gets you in and N gets you out.
That is the perfect summary. And understanding this 1-2 punch is how we created the antiviral drug Tamiflu.
Oh, really? How does Tamiflu actually work?
Tamiflu, or oseltamivir, is a neuraminidase inhibitor. It's a molecule that is shaped to look like sialic acid, and it gums up the scissors. It binds tightly to the N protein and stops it from cutting.
So the virus can get in, and it can replicate, but it can't leave the first cell it infects.
Correct. The new viral particles stay stuck to the surface of the dying cell. They clump up, and they can't spread to the rest of your body. This gives your immune system precious time to catch up and clear the infection.
That is fascinating. We aren't killing the virus directly, we are jamming their escape hatch.
Exactly. It's a tactical strike on their sugar processing machinery.
What about HIV? The text mentions a protein called GP120.
HIV is incredibly devious. Its surface protein, GP120, is the key that opens the door to our T cells by binding to a receptor called CD4.
Right.
But the problem with HIV from a vaccine perspective is that GP120 is heavily glycosylated. It is covered in a dense shield of sugars.
Sugars that are made by whom? The virus.
No, and this is the insidious part. They are made by us. The virus hijacked our own cellular machinery to cover its protein with our sugars.
So it's wearing our uniform as camouflage.
It's perfectly camouflaged. Because the sugars are self-sugars, our immune system generally doesn't see them as foreign. Antibodies tend to ignore them. And underneath that shifting sugar shield, the virus mutates the protein structure of GP120 rapidly.
So it's a moving target hidden under a friendly blanket.
This glycan shield is one of the main reasons why it has been so incredibly difficult to develop a broadly effective vaccine against HIV. The antibodies just can't get a good grip on the actual viral protein because of all the sugar that's in the way.
It's a wolf in sheep's clothing. If the sheep were made of sticky, constantly changing sugar.
A sticky, shifting heap. It's a brilliant, if terrifying, defensive strategy.
So basically, every major pathogen has found its own way to exploit this code. Malaria uses GPI anchors. H. pylori, the ulcer bacterium, binds to specific sugars in the stomach lining.
It's a constant evolutionary arms race. We change our sugars, they change their binders, we develop new lectins to spot them, they develop new shields to hide. It's been going on for billions of years.
This has been incredibly dense, but in a good way.
I hope so.
Let's try to synthesize this for everyone. We started by saying that sugar is much more than just fuel.
And I think we've proven that beyond a doubt. Glycoproteins are the ID cards, the anchors, the shields, and the communication arrays of cellular life. You simply can't understand modern biology without understanding the sugar code.
From the viscosity of the mucus that protects our lungs,
To the precise rolling of white blood cells that protects us from infection.
all the way to the sperm finding the egg, it's all run by sugars.
So for the listener, why should they care? I mean, aside from acing a biochemistry exam, what is the real so what here?
I think the so what is about understanding your own vulnerability and also your resilience on a molecular level.
Okay.
When you look at a disease like diabetes, you realize it's not just about energy balance. It's about preventing your body from physically gumming up with this age-accelerating chemistry. It explains why a tiny genetic error can cause a massive systemic disease like ICEL.
It demystifies things. It demystifies how medicines like Tamiflu actually work. It turns the abstract idea of disease into a mechanical reality that we can actually visualize and understand.
It makes us look at the body as an incredibly complex machine, but one that can be understood and potentially fixed.
Exactly. We are incredibly complex machines, and sugars are the gears and the sensors and the wires that make it all run.
I want to leave the listener with a final provocative thought to chew on. We touched on cancer very briefly in the outline, how cancer cells can use mucins to hide.
Right. Cancer cells are notorious for changing their glycosylation patterns. They produce what are called aberrant glycans, sugars that are different from healthy cells.
How does that help them?
Sometimes they overproduce mucins to physically mask their antigens from the immune system, literally hiding under a pile of slime. Other times, they change their surface sugars to help them unstick from the primary tumor and then restick to blood vessel walls in a different organ to metastasize.
So they're changing their sugar ID card to get past security.
Or to become more aggressive. So here's the thought. If we can crack the specific sugar kind of cancer, if we can identify exactly how a tumor's sugar coat differs from a healthy cell's,
Then we have a unique target.
we have a unique target. Imagine developing smart drugs, maybe engineered lectins or specialized antibodies that are designed to target only those specific aberrant sugars found on cancer cells.
So you could do what? Strip away the cancer's disguise so the immune system can see it.
You could do that. Or you could deliver a toxic warhead directly to the tumor, and only the tumor, based on its unique Schubel signature, sparing all the healthy cells.
The future of medicine might not be written in DNA, but in sugar.
I firmly believe the next great frontier in therapeutics is glycobiology. We're only just now beginning to scratch the surface of what's possible.
A sweet future indeed. Thanks for diving in with us.
My pleasure.
Catch you on the next deep dive. Stay curious.